Good morning. My name is John-Marc Chandonia. I'd like to tell you about Coral, a new data management and data analysis platform that Pavel Novichkov, Adam Arkin, and I built for the Enigma project. Enigma is a large environmental microbiology consortium between a number of different universities and national labs in the United States. Now, many organizations our size face challenges in managing and analyzing data, especially when the relevant data sets arise from multiple heterogeneous sources and methods. However, if this problem is solved, it opens up the kind of powerful analyses that just simply can't be done at a small single lab scale. Managing heterogeneous data properly has long been a grand challenge of data sciences, and the best practices have been formalized in these 4 FAIR principles. In practice, the last 2 are the hardest to achieve. Reusability is challenging because key details about many data sets are often implicit, not actually stored in the data files themselves. So once the postdoc who generated your data has left your lab, it can be very hard for your newer team members to fully reproduce or reanalyze older data sets. Interoperability, that is doing integrated analyses across data sets generated in multiple labs, is hard for similar reasons because different labs will document data to different standards of quality. Now, FAIR data has been achieved for some very specialized data types, including protein structures, evolutionary relationships between proteins, and genome sequences. However, doing this often requires a team of data scientists to handcraft detailed, complex data structures like these linked SQL tables that capture all relevant details of the data types. I call this centralized model the cathedral model of data management after the famous ESR essay. But as with cathedrals, building and maintaining these beautiful but massive complex data structures over time can be a huge investment. Most scientists would rather just store their data in unconstrained files, such as these Excel spreadsheets. This is often fast and easy at the time the data are produced, but this convenience comes at the cost of fairness because these file formats don't capture enough of the context of the data to make these data reusable or interoperable with other data sets from other labs across a big project. So in other words, current technology requires making these trade-offs between convenience on the left side of the figure, that is quickly developing, deploying, and maintaining new data types, and fairness on the right side of the figure, because the process of maintaining and deploying FAIR data models is very slow and expensive using current schema-based technologies. Now, how did we solve the problem with an Enigma? What we started with is we surveyed the breadth of work done by Enigma across the entire project to learn how to best model all of our data. And so what I'm showing here is just 1 year's worth of posters from the various labs involved across the project. Here's what we found. First, there are some key data types for which other groups have already thought very hard about how to model them. So we think that by building on these groups' work, the traditional schema-based process will be fine for us. But we have over 100 additional data types that for the most part look more like Excel spreadsheets. There's broad categories of these data types, like microbial abundance or growth or fitness or geochemistry or what have you. But within each of these broad categories, there's many subcategories. So for example, some data sets have replicates, some don't. There may be a time series or condition series. So what's the best way for us to build a data model for something like microbial growth that captures all these different flavors that are possible? So it turns out that fundamentally at their core, most of these new data types are basically just mathematical structures, things like graphs, trees, or for the most part, n-dimensional arrays. But an n-dimensional array is no different than a multi-dimensional Excel spreadsheet, no better that is. So we're still faced with the problem of how to encode the formal context that we would need to make these data actually fair. So to make the data fair, we invented this concept of the contexton, which we define as an atomic unit of context for data. So contextons are built using microtypes, which we define as an atomic data type representing a simple concept, such as concentration, temperature, or pH. By atomic, we mean types whose values would typically be stored as a single scalar. The names and definitions of these microtypes, their allowed units, and often their allowed values, are all defined on the space of ontologies, in order to provide a controlled, limited, and well-defined vocabulary. Now, a contexton is just an instantiation of one of these microtype variables. Contextons are required to provide formal context to the data that are actually stored in all the cells of an n-dimensional matrix, to each of the axes in the matrix, and to the properties of the data set as a whole. So together, these microtypes and sets of ontological terms can be thought of as a language that defines what data can be described in each instance of Coral. So for example, while all of the data in our Enigma Coral has the language concepts necessary to talk about Enigma data, a different set of microtypes and ontologies could be built to discuss medical data. So you could use Coral to track data in a COVID experiment, or even some completely different field like a financial data set. So the power of our system is that rather than building complex data types up front, Coral administrators just have to provide a set of microtypes and ontologies. This is analogous to a set of Lego bricks that, along with the mathematical data structures, allows users to build their own complex spare data types on the fly as they're needed. And so we call these data types dynamic data types. Coral stores both these dynamic data sets as well as traditional schema-based types, which we call static data, in a network. The network annotates the provenance of all data in the system. The nodes in this provenance graph are the data sets themselves, and the edges are a special type of static data we call a process. The process stores who did something, how, when, and why. This greatly facilitates adherence to all four of the FAIR principles. For example, users can find data sets by searching on the contents of the data, such as finding all data that contains uranium, or they can search using the network, for example, finding all the data that originated with a particular sample or sampling location. And because both static and dynamic data sets are built on top of a common set of microtypes, it's easy to merge linked data sets together with each other. And so Coral includes tools to do this automatically, which enables powerful integrated analyses of all the data stored in our system. If you'd like more information, I also have a short poster presentation, poster 445, and we filed the provisional patent on Coral, but we also hope to be able to distribute the code to you soon under an open source license, the license which will also allow free use of the patented technology for use in your projects. Thank you. I'd like to take any questions, please.